DNA sequences being deleted in an organism or inserted into the genome of an unrelated organism. To introduce new genes, a trait of interest with some benefit to the organism is selected. The gene or genes for this trait are then isolated, and the technique of comparative analysis is completed by comparison of the genomes with the trait to genomes of the same species. This identification technique determines where the desired trait is within the organism's genetic material. In order to replicate the desired gene, it is cut with the restriction enzyme and inserted into a new genome by one of the two methods. Gene gun technology is the more efficient gene transfer method used to introduce new DNA into living cells, organelles and tissues. Gene gun technology is more efficient gene transfer method used to introduce new DNA into living cells, organelles and tissues. The process uses DNA-coded particles which are propelled by the force of the gun through the desired organism cell walls, where the DNA is incorporated into the genome within the cells. Alternatively, the trait of interest is inserted into the soil bacteria Agrobacterium tumefaciens, which then infect the wound of the plant, injecting its DNA with the trait of interest into the plant, where it is integrated into the plant genome. Finally, the modified organism is grown in a controlled environment where the success and characteristic of the new genome is rigorously tested before the organism is multiplied and made commercially available.
One of the main limiting factors of canola is the significant impact that glyphosate has on the quality and quantity of the grain produced. The introduction of genetically modified canola has resulted in a huge profit for canola farmers, the market and the industry. Canola has been genetically modified to be tolerant to the herbicide glyphosate, resulting in Roundup Ready canola. Glyphosate is an active constituent in a range of herbicides used by farmers. The genetic modification enables glyphosate to be sprayed on weeds that are growing in Roundup Ready canola crops without the negative impact on the canola plants. Non-GM canola plants contain the enzyme EPSPS. This enzyme is involved in the shikimate pathway, which is a biochemical pathway that is important in plants. In this pathway, the enzyme is involved in the production of aromatic amino acids. However, when these non-GMO plants are sprayed with glyphosate, it inhibits their ability to produce the amino acids needed to grow and survive. GM canola was produced by inserting the EPSPS gene encoding the enzymes from the CPF4 strain of Agrobacterium and the glyphosate oxidase gene isolated from strain LBAA. Agrobacterium-mediated transformation method was used, utilizing single insertion event containing one copy of the tDNA from plasmid PVBNG-TO4. Only the DNA required to confer the glyphosate tolerance phenotype was transferred and inserted to a single locus in the canola genome. No genetic elements from outside of the right and left borders of the plasmid were transferred into or are present in the genomic DNA of Roundup Ready canola. Glyphosate oxidase enzyme accelerates the normal breakdown of the herbicide glyphosate into two non-toxic compounds, AMP acid and glycoxylate. AMP acid is the principal breakdown product of the glyphosate and is degraded by several macroorganisms, while glycosate is commonly found in plant cells and is broken down by the glycoxylic pathway for lipid metabolism. The main difference between conventional non-GM canola and GM canola is the gene technology that has been used to incorporate tolerance to the herbicide glyphosate.
Genetically modified canola is already grown in all other mainland Australian states. A moratorium in South Australia has prevented any form of GM crop production in the state, although as of August 2019, the state government is moving to lift the ban. The moratorium was initially established in 2004 and, unless removed, will remain until 2025. There are multiple arguments responsible for maintaining the moratorium, including GM-free label that supposedly gives South Australia premium grain prices compared to the rest of the country, and access to the premium international markets. Additionally, there is some concern for the possibility of gene flow into the outside environment, such as through the fertilization of nearby non-GM plants with GM pollen, which could create superweeds, where the GM plants contain resistant traits. Meanwhile, others argue that GM crops have greater environmental costs, particularly because of more chemical use. In actuality, these arguments do not have a firm basis. For example, the only evidence of a premium international market exists with Kangaroo Island growers and a high-priced Japanese market that stipulates being entirely GM-free. Provided that the moratorium remains for Kangaroo Island, it has been established that this trade may continue. There is also no data to support higher prices in South Australian grain simply from production in a GM-free area, while export data showed that South Australian commodities were not accepted any more readily into GM-free markets in the European Union. In terms of gene flow, it is uncommon between different species, and where cross-pollination of GM canola has occurred, it has been shown as limited beyond 5 metres from the plant. Hence, with appropriate crop management plans, the risk of gene flow can be well contained. Furthermore, the herbicide-resistant traits in GM canola enable better weed control with different herbicide groups, and consequently less chemical and softer mixes can be used. The moratorium has had a significant impact not only on growers in terms of limited herbicide rotation options and lost income, but the state of South Australia as a whole. The potential for GMOs in the future of agriculture means there is a significant amount of research and development focusing on GM crops and other biotech. However, as the moratorium heavily restricted growing GM crops even for research purposes, such research and development projects were not conducted in the state. This caused a decline in funding from domestic and foreign investors and loss of other benefits associated with these. The state's government is pushing to have the moratorium lifted by year's end, with the Primary Industries Minister claiming that lifting the ban on GM food crops will be giving the power of choice back to farmers. Key benefits will include better weed control and reduced risk of herbicide resistance from more options for in-crop herbicides, an increase in research and development investment in the state, and an overall reduced environmental footprint from reduced herbicide use. However, South Australian farmers will need to consider GM crops to remain competitive, and respecting a neighbouring farm's management practices just as conventional farmers do with their organic neighbours will become highly important.
To conclude, genetically modified canola, which contains unrelated herbicide-resistant genes, allows for better control of weeds and reduced herbicide use. A moratorium in South Australia means that GM crops are not currently grown in the state, but this will change if the ban is lifted, as the government is proposing. Although GM technology has seen much speculation and controversy, removing the moratorium will provide significant benefits for both farmers and South Australia as a whole.